Hi, in this presentation, we're going to explore the process by which seeds are going to develop. We're also going to look at the development of the embryo and also the process of germination and how that leads to the production of a new sporophyte plant. Now, remember that there are two kinds of plants, vascular plants, that can produce seeds. One is what we call the gymnosperms. Gymno means naked, sperm means seed, and gymnosperms include the conifers, like pine trees. And we also have the angiosperms, and the angiosperms are going to be known also as the flowering plants, members of phylum Anthophyta, and they produce seeds encased that develop, and then they are held within the fruit at least for a period of time. And so those are called the angiosperms because, as you know by now, angio means vessel or container, and sperm means seed. So we're going to be looking at seed development, embryo development, and germination from the point of view of the angiosperms. So what do we have in a seed? Let's take a closer look at this sketch, at this illustration. You're going to see that the seed is comprised of three main components. One is going to be the seed coat, and that is going to be there for protecting and enclosing the rest of the materials that are part of the seed, which are also going to include a nutritional material known as the endosperm. This endosperm develops from cells of the female gametophyte, and there's going to be an embryo, and this embryo is going to be developing from the process of fertilization, the union of an egg and a sperm. The seed coat, which is surrounding this entire embryonic system, is going to develop from the integument, which is in one ovule, and is going to be surrounding the female gametophyte. And so those are some of the connections we want to be keeping in mind as we look at seeds. And the rule, by the way, is that one ovule in an ovary is responsible for making one seed. And so we can see that in this seed, there's going to be two places where nutrition can be provided to the developing embryo when germination takes place. One is the endosperm, but also part of the embryo is going to include these structures called cotyledons, from where we make names like monocot and dicot. And there's going to be nutrition over here too. With regards to the development of the embryo, this illustration can help us make some of those connections I have just mentioned at the beginning of my presentation. The zygote, as you know, is going to be a fertilized egg. The union of the egg and a sperm produces a zygote. First, this zygote is going to divide by mitosis and make two cells, a terminal cell and a basal cell. Let's see what develops from the basal cell. It is going to be a structure known as the suspensor. This suspensor is going to be multicellular, and it attaches the rest of the embryo to the parent plant, and it's going to be a vehicle through which nutrition is going to be passed from the endosperm to the embryo as well. The other cell we have here, called the terminal cell, is going to be forming most of the embryo, which is contained in the seed. First, a proembryo forms, which, as you can see here, is kind of shaped like a balloon, and that is going to be because two structures called cotyledons will be forming. Eventually, other embryonic structures will also be forming as the embryo gets longer. That's what we mean by elongating. The seed is going to be something that is used by plants to propagate, to spread out. It is important for the plant to disperse in this fashion, because if all of the seeds landed right there next to the parent plant, there will be a lot of competition. And the seed coat is important because, remember, it's going to be protecting the embryo and that nutritious material that this young plant is going to need. Seeds are going to be, once they are finished producing, they're going to be entering a state of dormancy. During the state of dormancy, the content of the seed in terms of water is going to be only about 5 to 15% water. So a seed is something that is relatively dry, and that is okay because the metabolic levels within the cells of the embryo are going to be dropping also to a minimum. And something about the dormancy of the seed is a benefit because seeds want to germinate only when conditions are favorable. And so there are different factors that are necessary for breaking a seed out of dormancy. One of those is going to be like an abundance of water. In places that are dry, it takes a lot of water before a seed can germinate. And so just a little drizzle is not going to activate germination. But it usually takes a lot of rain, even in dry places, there's going to be like a wet period, a rainy season, and that's when development is going to take place. Some seeds actually require high temperatures, like a fire must burn through an area before seeds can germinate. Other seeds require going through the digestive system of an animal where the gastric juices, the hydrochloric acid, helps break some of the seed coat, that protective layer, before a seed can sprout or germinate. So let's take a look at the embryos of monocots and eudicots. And now we have this illustration I'm going to be using. So remember that most of this embryo material here is something that developed from the terminal cell. The basal cell helped form this suspensor, which attaches the embryo to the rest of the plant. So when you look at the embryo, you're going to see that there's going to be a first set of leaves. There's going to be an epicotyl. Epi means top. And there's also going to be a hypocotyl. This epicotyl is going to be a place where an apical meristem is going to be found. And the hypocotyl eventually is going to end at a region known as a radicle, where we have another apical meristem. This radicle is going to have a tip where the root of a new plant is going to develop and grow. Now, if we look at a monocot, we're going to see some of those same structures. We're going to see, here's the embryo. This is going to be the first leaf. This is going to be the epicotyl on top, hypocotyl on the bottom, a radicle where the roots are going to be forming as this seed germinates. One detail that I wanted to mention about the monocot seed is that the embryo is going to be protected by two shields, and one of those is going to be the coleoptile that is going to be protecting the plumule right here where the apical meristem, the first leaf, and the epicotyl is going to be. When the seed sprouts, sometimes it's going to be pushing through hard soil, rocks may be present, and so this coleoptile will be preventing this initial shoot from being damaged as it breaks through the ground. And here the root is going to be, or the radicle, we don't have a root yet, the radicle is going to be protected by another shield, which is known as the coleorhiza. The prefix of these words, coleoptile and coleorhiza, is something hard. It's a hard protective shield. Now, if you look at the cotyledon, monocots only have one cotyledon. That's why they're called monocots. And it is also going to be in the form of a shield. It's going to take that shape, and it is also known as the scutellum. So here we can see in this illustration the structure of the embryo in a eudicot seed and the structure of the embryo inside of a monocot seed. Let's focus a little more on the eudicots. Just wanted to mention the fact that in many of the eudicots, the nutritious material is going to be kept inside of the cotyledons. And what happens is that the endosperm of these embryos, surrounding these embryos, those are going to be passing most of the nutritious material to the cotyledons, and so the endosperm practically disappears. So in case you're looking at this illustration, you're looking at, here's a bean, we see the cotyledons, but we don't see the endosperm, and the reason is the endosperm resources, most of them are taken up by the cotyledons. Now you can see some eudicots, like this castor bean. The cotyledons remain, but they're going to be thin and slender, and again, most of the nutritious material is going to be right there in the endosperm still. Okay, so that's a detail I wanted to mention here. Let's talk about seed germination, because we just mentioned that a seed is going to be relatively dry, like 5 to 15% water. The main requirement for a seed to sprout, to germinate, is going to be taking in lots of water. And so that's a process known as imbibition, which is something that for people who are familiar with the consumption of alcohol, imbibing, it's a word used sometimes when people say they're going to go and drink alcohol. So imbibition, but in the case of the seeds, they're drinking water. Okay, let's make it clear. They're underage. The plant's just beginning its life. The radicle is going to be always a part of the embryo that emerges first in both the monocot seed and the eudicot. The radicle emerges first. And it's going to have to be breaking through the soil, breaking through sometimes hard substrate, so it can get the plant anchored. That's one of the advantages for the radicle coming out first. The plant needs to be anchored. And number two, remember that the radicle will lead to the production of a root. And so because the plant will need to drink lots of water, a root is going to be beneficial for the plant to obtain this precious water. Now, when you look at the next events that take place, things will be a little bit different with regards to the eudicots. So here we see the radicle coming out first, and then the hypocotyl is going to break out, and it forms a structure that looks like a hook. This hook here is going to be breaking through sometimes hard soil. Remember, sometimes there are rocks here, and because it is a tougher type of structure compared to the plumule, the apical meristem, the first two leaves of the embryo, this is going to be breaking the ground. Eventually, the rest of the seed comes out. At the same time, the root is growing. You can see a primary root. You can see some secondary roots that are beginning to emerge. And after the entire seed goes out from the ground, then the foliage leaves grow out. The cotyledons do their job of providing nutrients to these foliage leaves, which begin to do photosynthesis almost right away, but there's going to be a lot of need for energy. And so while the plant's beginning to do photosynthesis, they continue to rely on the nutrients from the cotyledons. Eventually, the resources here are depleted, and there's no more nutrition, so the cotyledons are going to shrivel, they dry up, and they just fall to the ground. Meanwhile, the rest of the root of the plant has continued to anchor the plant firmly to the soil and provide the much needed water for photosynthesis. Now, if you look at the emergence, the development of the monocot seed, here's the radicle, which comes first. Next comes the coleoptile. We don't see that hook, the hypocotyl forms, because in this case, remember that the coleoptile is like a shield that is protecting the plumule, protecting the first leaf and the apical meristem and the epicotyl, and then this is going to be what's breaking the soil, the ground, pushing away small pebbles. And once the coleoptile has emerged, then eventually the first foliage leaves are going to emerge. And again, the growth of the root continues so that more water can be provided for photosynthesis and the plant can be safely anchored. And that is how seeds are structured. That's how the embryos develop and how germination takes place in the angiosperms, the flowering plants.